So Mendel actually, he self-pollinated that. We saw up to F2, right? So first he took 2 pure lines and then they form a heterozygous, and if it is this is the F1 generation, and he self-pollinated these 2, that is the heterozygous, and we got the different ratio in the F2 generation, right? So he self-pollinated F2 and found that dwarf F2 plants continue to generate dwarf plants in F3 and F4. So again, he tried with the 3rd and the 4th generation, and he found the production of more dwarf plants, right? So he concluded that the genotype of dwarfs was homozygous. So the dwarfs were expressed only in the homozygous condition, right? That is small p, small t. And apart from studying these height, he also observed on the other traits like flower color, the part color. So all the other contrasting traits also he studied. And he found that some factors were inherited from parent to offspring. So when he studied the different generations, he found that some of the factors are inherited from parent to the offspring. And these factors are now called as genes, right? So what he observed as factors is now called genes. So what do you mean by back cross and the test cross? That is, we saw that as a result of the mathematical explanations, we cannot find that whether the plant is a homozygous tall or a heterozygous tall. So that can be found out by means of a test cross. So what is a test cross? It is the cross of an organism or crossing of an organism with dominant phenotype to a recessive individual. For example, we are crossing only with the recessive individual, that is with TT. With the dominant phenotype, it may be either TT. For example, if TT, that is homozygous TT, is crossed with the TT, so what will be the offspring that is produced will be, everything will be tall, right? So when you put the Punnett square, you can easily find out that this T will, this is the F1 generation that is produced. All will be of the heterozygous type. But if this heterozygous parent is crossed with the recessive parent, so here what will be the ratio that we will get? So if TT is crossed with TT, so first it is TT, and here it is TT, and here it is TT. So what is the ratio here? It will be, that is in the ratio of 1:1. That is 2 tall and 2 dwarf. That is 1:1 ratio. If it is a heterozygous parent, right? So we can find by means of the test cross, we can find out whether it is a homozygous tall or a heterozygous tall. Okay, clear? So that is the test cross. So crossing of an organism with dominant phenotype to a recessive individual. And what do you mean by back cross? It is a cross between a hybrid and its any parent. So with the hybrid, and if it is crossed with the any parent, then it is called a back cross. So test cross is used to find out the unknown genotype of the character, right? Example, flower color or any type of character we can find out. So back cross and the test. So here we are taking the homozygous recessive, that is WW. This is a recessive, and if it is a, we are not sure about the phenotype of this parent, right? So here it may be either WW or it may be a heterozygous. So if it is the homozygous, here all the flowers are violet. The same thing we saw as the previous slide, right? So here we took the example as the tall and dwarf. So here they took the example as the violet flower, which is the dominant character, and white, which is a recessive character. So here, if it is a homozygous type, then all the flowers are violet. So if it is a heterozygous type, half of the flowers are violet and half of the flowers are white. So then if the flowers, all the flowers are violet, then we can conclude that that flower is a homozygous dominant. And if that unknown flowers after first the one generation, if we find violet and the white flowers, we can conclude that the flower is a heterozygous flower. Clear? So this is the basis for S cross.